Our last speaker of this session is Zach Serber from Amyris.
Good afternoon. So one of the consistent themes of this conference so far has been the egalitarian nature of synthetic biology. It really seems very plausible that the next amazing breakthrough in our field might come from a high school student, and that is really quite something. That can't be said about most scientific disciplines. And that's partly because it's so fresh and new, and we're trying to make it available to everyone. Another theme I'd like to endorse as part of synthetic biology is the value of taking our ideas and commercializing them, making products with the things we develop in the laboratory, whether it be academic or in a competition, it doesn't matter. There is a lot of potential for commercialization of these ideas. And Amyris is one of the companies that started trying to make a difference using these technologies. And this talk has been titled Out of the Lab and Into the Streets to emphasize that idea, that we took some ideas that were developed in the Kiesling lab, added to them, expanded them, brought in a business team, and are now finally launching our first product and are well on our way to multiple other products. Now, I'm going to be telling you a couple of stories out of Amyris, some updates on the news. And I think probably the best news out of Amyris lately is we just finished raising money. We're not out looking for money right now, which is indeed fortunate. But the science is going really well also. So the initial funding that we received was from the Bill and Melinda Gates Foundation to the tune of about $40 million to pursue treatments for malaria. Now, malaria is, as you know, transmitted by mosquitoes, and it destroys red blood cells and has a tremendous health and economic impact around the world. Now, there are a bunch of older drugs that have been used to treat malaria, chloroquines being the most notable examples. The problem is that Plasmodium parasite is now resistant to chloroquine around the world. So here in yellow are areas of the world where malaria strikes, and the dots indicate various kinds of drug resistance that have emerged. Basically, the chloroquines more or less don't work anymore. Fortunately, there are other, and I dare not call them new drugs because they're actually very old drugs, but there are other drugs that haven't been commercialized at the same scales that are available now, to which drug resistance hasn't been developed in any serious way yet. Artemisinin, developed or extracted from sweet wormwood, is one of these candidates. So here's a brief history of artemisinin, and it's very appropriate to be giving this presentation here because the earliest discovery of artemisinin was in China and was discovered in 168 BC as a treatment for a bunch of things, including hemorrhoids. It was also discussed in a text in 340 AD as a treatment for fevers and malaria. In 1972, the active ingredient out of Artemisia annua was identified as artemisinin. So, malaria causes between 1 and 3 million deaths per year, and the majority of victims of this disease are the very young and the elderly. Treating malaria worldwide would require something between 300 to 500 treatments, given the death rate. And that would require about somewhere maybe 300 to 400 tons of artemisinin, which, given its current source, which is plants, would require 6 million tons of plant material. And one of the problems with deriving it, aside from the sheer volume of plants you need to generate enough material, is that it's intrinsically expensive to extract it, and the supply of the plant varies according to drought and flood cycles, and has fluctuated. A kilo of artemisinin derived from plants has fluctuated in the last 5 years from $300 a kilogram to $1,200 a kilogram, just depending basically on the amount of rain the countries that produce it have received. One of our objectives is to flatten that supply. So the traditional method is to cultivate the plants, which takes about 2 years for seedlings to grow up to maturity that they can be harvested, and then to extract the drug from those plants. What we proposed to do was to take the relevant enzymes out of the plants, put them into E. coli and later yeast, and ferment those microbes whose sole purpose in life, as far as we're concerned, is to make artemisinin, or artemisinic acid, actually a precursor, and then to use chemistry to take it from artemisinic acid to the final product, artemisinin. And this would become part of the ACT therapy. So the original project, as it was initiated in Jay Kiesling's lab, was to take E. coli and amp up the mevalonate pathway by taking some genes out of yeast, putting them in to generate more IPP, and then also taking a pair of genes from Artemisia annua, the amorphodiene synthase, and the P450 associated with production, and put those into bacteria as well. When you do this, without very much in the way of optimization, you can generate, after some iterations, something like 500 mgs per liter, which is pretty good for a first go. But as we saw earlier today, there's a lot to be gained by taking that first starting point and optimizing around it by varying sources of enzymes and promoter strength and things like that. And we've taken a very similar approach. So we begin with acetyl-CoA in the pathway. We have a bunch of genes extracted from yeast, the MEV pathway, the top part, and then the bottom part, which takes us, plus an additional gene, to FPP. Amorphodiene synthase takes us to amorphodiene. Then the P450 and the CPR take us to artemisinic acid. Artemisinic acid is then purified from the bugs and then goes through some chemistry steps to make artemisinin. One of the interesting parts of this project has been trying to bring the cost of this chemistry down as low as possible, which is not something that most synthetic biologists think a lot about, but is definitely part of the bottom line and the feasibility of a project like this. And when I say bottom line, I should emphasize from the start, this has entirely been a non-for-profit project. Amyris is not going to make a dime off of this, but we have gained tremendous insights into how to do synthetic biology on an industrial scale. And those insights we're bringing to bear on our fuels program, which I'll talk about in the second half of this presentation. So artemisinin can then be used as a starting point for a series of derivatizations that can make a series of other drugs that all can be used to treat malaria and perhaps some other diseases as well. So in terms of our first attempts to optimize it, we started with the top pathway, which was under a PLAC promoter, and put under a LAC-UV5 promoter and codon optimized each of these genes, which in the first iteration wasn't the case. And if you do this simple step, what you see is we go from making about 500, 600 mgs per liter to up to about 7 grams per liter. So that was a pretty huge boost just based upon a simple optimization. We then started changing around and going after different enzymes for different organisms. Enterococcus faecalis, which came up in the last talk, was a source for a couple of genes. We also went to Staph aureus for some genes on the grounds that there seemed to be some optimization to be done. There wasn't a big difference here, but we could get, this is done in shake flask rather than a fermenter. We found that the Staph aureus seemed to give us a hit. We then went back to Staph aureus and said that maybe we could make some success by adding the synthase in that position from Staph aureus. And indeed we did. We saw an improvement, and we had amorphodiene yields up over 15 grams per liter pretty quickly. Now this was the first process we tried. Another thing we've learned is the tremendous value that is to be had by optimizing your fermentation conditions. There's a lot you can do with a pre-existing bug by simply changing its growth conditions. And that same bug was taken through a series of optimization into a process I'll call C. And this is triplicate runs showing the reproducibility of it. And now we're getting over 25 grams per liter of amorphodiene. So if you look at the time, so I should say that one of the goals of the company was to reach 25 grams per liter. And if we plot that progress over time, it looked like this. Now this long semi-plateau region was largely to do because once we had reached the goal, we stopped trying very hard on this objective. And we redirected our efforts elsewhere in the project. But we still dabbled in it, and we've now gotten up to about 43 grams per liter of amorphodiene, which is pretty sensational. Now, that's amorphodiene. That's not the end of the pathway. We're taking it to here. We want to get to here using the CPR, the cytochrome P450 reductase, and the P450. And it was around this project that we started to explore other host organisms. Up until now, everything I've talked about has been in E. coli. We decided to consider yeast as well because there has been a lot of precedent around using P450s more successfully in yeast. And indeed, that seemed to be the case for us as well. One of the things we uncovered very quickly, though, upon moving to yeast, was the yeast seemed really stressed out. We couldn't generate as much artemisinic acid. So this is equal molar amounts of amorphodiene here. This is arbitrary units, versus the amount of artemisinic acid we made. And when we probed these yeast using some of our omics platforms, and we have a bunch, we discovered that genes associated with oxidative stress were being upregulated or downregulated accordingly. The pattern was very clear. This is not so surprising. If you look at the reaction mechanism of a P450, one of the things that naturally can occur is a generation of reactive oxygen species through an unintentional decoupling of the CPR from the P450. And even evolutionarily optimized P450s in mammals often have a 50% incidence of decoupling and generation of reactive oxygen species. And it probably was the most likely source of our problems. We decided to use a rhodamine dye that was reactive with oxygen species to glow green as an assay for this phenomenon, because to try to solve the problem, so as to get artemisinic acid yields comparable to amorphodiene titers, we had to understand what was going wrong. So we spent a considerable amount of time trying to diagnose this. And here we see, this is a flow cytometry where the further left you are, the less green the cells are. The further right you are, the more green the cells are. And here is strains that are only producing amorphodiene, and basically there's very little in the way of reactive oxygen species in these cells as measured by green fluorescence. When you go all the way to artemisinic acid with the P450, you see a substantial population of the cells shifting over into the green regime, which indicates they're undergoing generation of oxidative species. Now, I'm going to have to abbreviate a little bit here, but suffice it to say that we discovered a solution to this, and we now can generate artemisinic acids at comparable levels to amorphodiene. Oh, yes, and just to show that cells with high fluorescence here, this is basically a numerical depiction of what you just saw, and the correlation I want to draw here is viable cells. The least viable cells also tended to be ones that had the most green fluorescence, implying that green fluorescence and reactive oxygen species were also associated with unhappy cells, unviable cells, and the ODs don't grow as high either. These cells are sick. So we solved that problem. Let's skip through that. And around the time we were first confronting these problems, we announced a partnership with Sanofi Aventis over in Europe to go into full-scale production of artemisinic acid and ultimately artemisinin. One of the things we've learned as a company is how to negotiate these sorts of deals and to work with partners to make something commercial. We don't have necessarily the expertise in-house to make millions of tons of something. We are still a relatively small biotech company. We develop the technology, but we would rather have experts actually carry through and make the product. And Sanofi also is going to treat this as a nonprofit project. They are not getting a dime from it. And they are investing $20 million of their own into CapEx, basically, infrastructure around the particular idiosyncrasies of fermenting these bugs and doing the chemistry around making artemisinin. So that is our first product, and this is a tremendous success for us. We still have scientists in-house working on this project, but they're fewer and fewer every day because basically we've passed it on to Sanofi. There's always more to be done. As any graduate student will tell you, you're never quite there, but by any measure of commercial success, we're there. Now, I keep on referring in passing to what we've learned from this process. Well, basically what we did was we took glucose, engineered a microbe, both E. coli and yeast, to make amorphodiene. Amorphodiene is a hydrocarbon. Could it be a good fuel? Well, not all hydrocarbons are good fuels, but some are. So let's consider a very abstract notion of what we might go do next. Well, we can create a Venn diagram, with one category being molecules with good fuel characteristics. And these are defined by things like government standards as well as emission profiles. Then we should consider molecules that work with today's infrastructure. And not all molecules that have good fuel properties necessarily work with today's infrastructures. And I would argue that conventional biodiesel would fall into that category, and maybe ethanol as well. Around issues around material compatibility, refining, and transportation. And finally, we have to consider what biology can readily make. And so in this intersection here would be conventional biodiesel and ethanol, molecules with reasonably good fuel properties, and what biology can readily make. What we do with synthetic biology is basically expand the scope of what we can conceive of making with biology by mixing and matching parts routinely. And what Amyris did was basically go explore this space. What can we make with synthetic biology, which is both going to be a good fuel and work with today's infrastructure? Now, I kept on mentioning ethanol and fatty acid methyl ester biodiesel, and they're a very good start, but they do have issues. In the first place, ethanol has material compatibility problems. It has a high vapor pressure, high water solubility, and low energy density. And some of these, like the water solubility issue, means that you can't transport it through pipelines, for example. Low energy density means you have to put more in your tank to drive a mile. Conventional biodiesel has a high cloud freezing point and poor pipeline transportation ability. Now, you can make these work, but you have to do things to the infrastructure to make it scalable. And anytime you have to change something as large as the infrastructure around transportation fuels, that comes at tremendous cost. And the likelihood that people are going to adopt that new technology drops precipitously. We wanted to generate products that people could use immediately without compromise. Another thing we wanted to do was take advantage of everything that had been learned in the process of making ethanol. So one of the things the Brazilians have been doing very well for 30 years is taking Brazilian sugarcane, taking it to a mill, and making lots of ethanol with very low carbon footprint. This seemed like a good idea. We just wanted to expand their scope to consider things other than ethanol, something that had better fuel properties. So here, these are going to be some slides about which I am not expert. These were generated by chemists at Amyris doing a variety of tests. But what we're going to be comparing here is some of Amyris's next products, one of which is our main product at the moment is our Amyris diesel product. Across the top, what you'll see here is conventional diesel. In the middle, this is soy methyl esters, basically a kind of conventional biodiesel. And at the bottom, Amyris diesel. And we can just look at different categories. So the cloud point is a point at which the temperature at which particulate matter starts to come out of solution. And basically the reason why you can't use conventional biodiesel in any place that gets very cold, because it will clog up your engine. Amyris biodiesel has a cloud point below 50 degrees Celsius. The cetane number is a measure for diesel, kind of like octane. And Amyris scores a little bit better than conventional diesel and better than soy methyl esters. And then in terms of energy density, we're very competitive with conventional diesel. Now, this is going to be our first fuels target, principally because it is the fuel which is going to make the biggest difference on the world, the fastest. Diesel is the fastest growing energy transportation fuel, and people from the United States and Japan tend to think that the world runs on gasoline, and it just ain't so. The majority of vehicles in the world do run on diesel, and this is where you can really make your first difference. Our second product, which we're also working on aggressively, is our jet fuel molecule. Now, jet fuel comes in second because really there isn't at the moment any kind of renewable fuel alternative to conventional jet diesel, except, well, it's not renewable. This is conventional jet fuel. This is coal-to-liquid technology, which you might have heard about in some political spheres. The Bush administration has been a big fan of this. Basically, through an elaborate process, you can take coal and make a liquid out of it that you can burn in a jet. It has horrible greenhouse gas properties. And then Amyris is jet fuel. So in terms of energy density, we are the most dense, but it's basically comparable. The freezing point of the jet fuel, which is obviously important when you're flying at 30,000 feet, is below 70 degrees Celsius. And the greenhouse gas emissions for our Amyris jet fuel derived from sugarcane is going to be less than 80% that of conventional jet fuel, using conventional jet fuel as a benchmark. And finally, our final fuels product is going to be a gasoline equivalent. I'm just going to go through this quickly. We're comparing it to ethanol. Here's our biogas, and here is conventional gas. The vapor pressure is very low. the energy density is a little bit higher than conventional gasoline and a lot higher than ethanol. Ethanol is not energy dense at all. And the octane, well, you know, ethanol has a tremendous octane. Did you know that? It's interesting. And we're pretty standard around with conventional gasoline. So here's a picture taken 11 months ago of E. coli, the little rods here, producing our diesel product. Those are the droplets there. I was there when this photo was taken. This was undoctored. It's true to life. And more recently, it's become evident that there's a lot to be gained by making fuels in yeast rather than E. coli, particularly if you want to be compatible with existing infrastructure used to make ethanol. And so here's a picture of yeast making another droplet of our fuel, taken more recently. This was in April. And one of the nice things about our fuel, as opposed to, say, ethanol, is the purification. Ethanol has to undergo a distillation process to separate it from the water before it's put into an engine, and that's a very energy-costly process in and of itself. Our product, the diesel, just floats to the top upon low G centrifugation. No distillation is required. And so what you get after a crude recovery by centrifugation is something that's 98% pure, and the vast majority of the contaminants are also other hydrocarbons, the implication being that those can probably also be burned. You may not even have to remove them. That remains to be seen. If you do additional purification, the color goes away. It looks less like LB and more like fuel. And you're ready to put it in your engine, pretty much. So this is a movie of the separations going on, and it tends to crash my computer when I run it here. So once I get to the end of the talk, I'll run it in another window, just in the background. It's kind of fun. Here is the centrifuge here, and what you'll see is dripping in is the diesel, that cloudy LB-like stuff, and here the cell waste and the water. So a little businessness to this. I actually cut out a lot of these slides because I can't speak intelligently about them. But we used to be in this space. We used to be 50 people around the time I joined, and now we are 203 full-time employees as of Wednesday. We have this great new space, which is just below J-Bay. And we have raised over $100 million in venture capital and grants from a variety of sources. A lot of prominent VCs are involved. And we have lots of things going on in biology, which I know the most about. But we also have lots going on in fermentation, downstream purification, and we have pilot plants under construction. Well, actually, our pilot plant in Emeryville is going to be running this coming week, and one in Brazil as well. Here are pictures of our pilot. This is a picture from the biolabs to the pilot plant across the street. It's in this warehouse here. These pictures are actually a little old. This is August, and in business time, that's ancient, because these labs are actually finished now and have the fermenters in them. And we're going to run them for the first time next week, and we're going to make about 500 liters of beer as a test run. So as I keep on mentioning, and it probably goes without saying, that our focus is on sugarcane. We do believe in cellulosics, but we also think it's not there yet. We are feedstock agnostic. If someone out there in the audience can please develop a way of converting economically cellulose into a feedstock, we want to work with you because we would happily use that as our feedstock. But at the moment, the most environmentally sound way of generating the carbon necessary for the fuel through photosynthesis is using sugarcane. And we have recently formed a joint venture with Cristalsev in Brazil, which is the second largest producer of sugarcane, to get that process going. And they've made all sorts of commitments to us and us to them, and we are going to be making diesel in their fermenters as soon as possible. And so we're focusing on the business side. Again, this is not my expertise, but on a very broad scale to various aspects of the business process, from working with growers to generate the sugarcane. We want to make our process completely compatible with existing infrastructure so you don't have to make special fermenters just for our bugs. And also working on making it so that the fuel is obviously cost competitive. We don't want this to be a luxury item. This is not going to be a yuppie fuel. This is going to be the most cost-effective fuel you can buy. And that can be used in existing engines with no problem and transported through existing pipelines with no problem. And the estimated life cycle greenhouse gas emissions for our fuel is going to be about reductions of 80% as compared to petroleum products. Basically, of course, this goes without saying, the carbon we're putting into the atmosphere when you burn our fuel is comparable to fossil fuels, but that carbon came from the atmosphere in the first place because it was fixed out of the atmosphere by the sugarcane in the first place. And here is a bunch of people on our R&D team, business team, and outside directors. I want to emphasize, when you want to go commercial, you got to have a pretty strong business team, and we have not neglected that part of our business. And so that's the end of the talk. I'm just going to run, as I said, the movie in the background, and I can take some questions. Okay, questions?
Thanks a lot. So Chris.
Chris?
I know you can't talk about which molecules you're working with, but at different points in the talk, you mentioned jet fuel, biodiesel, Amyris biogasoline.
Yep.
Can any of those products be put into my car right now? I drive a 91 Nissan Maxima.
So our last product can. So you wouldn't want to put our diesel product into your gas tank, nor our jet fuel product. But you can certainly put our biogasoline product into your car. That is the whole idea.
Can you put your diesel into a biodiesel or a diesel car?
Yes, absolutely. Yes, I'm sorry if that wasn't entirely clear. The whole point is that you don't, as a consumer, have to change anything. The way you'll get the most rapid and aggressive adoption of a new technology like this is if the consumer can be ignorant of that there's even a difference. If the consumer can go to the tank and say, oh, it's less for Amyris's product, and it does exactly the same thing. In fact, we have a slightly higher energy density, so you can go a little bit further on that gallon. Well, let's just put that in because it's cheaper. You can mix it with gasoline.
It's 1 year, 2 years, 10 years.
Into what?
Time frame.
Time frame for commercialization? To put in your car. Well, let's say for the sake of argument that you drive a diesel car, because I can speak we're closest to the diesel products so that I can be most accurate about that. The intention, according to the timeline, which is to launch the Amyris diesel in the early part of 2011. We probably will have a little bit of it in Brazil. So one of the things about sugarcane manufacturing, which is kind of interesting, is the sugarcane to ethanol business is you have an ethanol plant here, and then you have sugarcane grown in a circle around it that's about 80 kilometers in radius. The reason why you have that is because the cost, both financially and in terms of fuel, of transporting sugarcane to the mill is a limiting factor. And those are done by trucks, diesel trucks. So the ethanol that's produced in the mill cannot be used to power the trucks that actually transport the sugarcane to the center. One of the first applications of our fuel, which will be before 2011, will be to power the trucks that transport the sugarcanes to the center of the factory, the center of this little circle, big circle.
Yes. Sorry. What?
So the more energy you convince a microbe to put towards a fuel, the less it can put towards its own growth and metabolism.
Absolutely.
So are you able to talk about any strategies you can use towards reducing the accumulation of mutants? Do you have to throw out all your accumulated biomass after a certain number of cycles and start fresh? Or what do you do to counter that?
That's a very informed question. It's a very good question, and one that my group directly addresses. And yes, there is, when you need to make something cheaper than it takes to pump it out of the ground, every ATP counts, literally. I mean, we have a conversion of ATP to cents. We know if we waste 1 ATP, what that will cost us in terms of the final gallon that's sold at the gas tank. So we need to be ultimately maximally efficient. Biomass accumulation is a big energy suck. And so if you can use your biomass again and again, and this is what they do in ethanol production in Brazil currently, is they will actually use the same biomass to produce the ethanol for months. And we certainly are inspired by that, let's say.
Okay, Kim, do you have?
Yeah, you mentioned that your biodiesel there looked a lot like Albi, and in the video that you had there, it looked like basically a bottle of growth medium.
Mm-hmm. It's not, I promise.
Does anything grow in it? I mean, do you need to put antibiotics in it so that you're not going to end up with contaminated fuel?
That's a good question. I don't, probably someone's looked into that. We're 200 people. I don't know. I really doubt anything's going to like being in there. However, life is very diverse, and life finds a way. No, I'm not especially worried because, frankly, we're not going to sell it like that. As soon as you get it off the centrifuge, you're going to take that and take it into a polishing step, which goes through an additional kind of purification, which I can't talk about, to make it the clear stuff. And then that clear stuff is what's transported in the trucks.
Okay, right there in the middle.
Here.
Yes?
So, how much first does it cost now to make a gallon of that fuel? How long does it take to make a gallon? And how much biomass do you need?
I can't answer any of those questions. I'm sorry.
Okay, moving on.
Yeah, sorry.
Blue shirt here.
Okay, phrase it another way. There's been a great debate about, food production and biofuels. What's your company line on this? Because it's quite controversial, certainly in the country I live anyway.
Yeah, absolutely. And we are very obviously aware of the debate. And how do I say this? I mean, the company was founded by a bunch of people who were working for substandard wages to work on a cure for malaria for starving children. Like the cultural foundations of the company is, despite the fact we have a corporate team now, not very corporate. Like we are a lot of do-gooders, and we're very sensitive to that. And so when I say this, I'm trying to basically think. I mean, I'm sure my judgment is clouded somewhat, but.
Well, I guess what I'm saying is in 2011, how much of the world will be covered in sugarcane?
Right. Yeah. So. Yeah, I mean, one of the, okay, yes, so the sugarcane is being grown in Brazil in basically the southeast portion of it, which is prairie land. It's flat. It's not near the Amazon. It's mostly pasturage for cows. Like one of the things I learned being at Amherst is one of the best things you can do for the environment is go vegetarian, because of the energy cost of making meat is so much greater than making plants. So one of the strategies for expanding sugarcane production would be to encroach upon the pasture land in Brazil. So each cow in Brazil currently has 2.5 acres devoted to it, which is quite, certainly by American standards, luxurious, where everything's in feedlots. And I'm not necessarily going to advocate a feedlot strategy, but you can make the cows more dense even without going to a feedlot situation. There have been calculations certainly on the amount of acreage you would have to cover with sugarcane to generate enough biodiesel, for example, to run the world. And depending on your perspective, it's not a lot or not a little, but I can't remember the numbers offhand. I mean, it's certainly less than the square acreage of southern Brazil.
Okay, we're not going to solve this problem right now, I don't
Right.
think. Was there one question here, right here? Or, sorry, blue back there.
Hi.
Oh.
I wonder if there's a membrane integrity problem when you have so much hydrocarbon in your medium.
Apparently not.
So did you check the viability of the strains?
Oh, yeah. I mean,
and it is high.
Yes.
Yes. No. Okay.
I mean, most of the time. I mean, there are things that we do, but that don't have to do with the fuel production that impact viability.
Now.
Thank you. Just to come very quickly to this guy's question. Tomorrow on the Global Social Impact panel, Camila Moreno from Terra de Ritos, who monitors sugar production in Brazil, is going to be speaking about the impact and particularly how new sugar production is actually pushing into the Amazon. So it's not entirely true what you're saying. We'll talk about that tomorrow. I'd be interested to know, what are the biosafety measures in place at your Emeryville biorefinery? How do you prevent your microbes, your yeast that might produce gasoline from getting out?
Right. So basically, I mean, I can say a lot of things about that. The first thing I should say is kind of generic, but we are certainly obeying every regulation and law in the process of making that plant and cultivating those yeast, which include, proper disposal of the biomass that's produced and air filters and everything else that's associated with it. But I will also add that from a scientific point of view, our yeast wouldn't last a second out there in the world. I mean, one of the things we talked about with biomass accumulation, is basically a fuel or biomass issue. And if you're making fuel aggressively, you're not making biomass, and from an evolutionary point of view, that's a bad place to be.
Okay, I'm going to use prerogative of the chair here, and I want to ask one different question.
Uh-huh.
What's the target of artemisinin, and what's your sense about mutation rate for resistance?
Right. Right. So actually, the the World Health Organization identified the first example of some resistance to artemisinin, I think in the Philippines, about 6 months ago. So there has been one documented case of it so far. This is a very important issue, and one of the things that the World Health Organization advocates is a combination therapy of artemisinin with other very effective drugs as well. It's just one of the ingredients. One of the worst things that can happen, and has happened, is using artemisinin, or basically unscrupulous manufacturers selling artemisinin that contains a tiny bit of artemisinin in it so it can fool the mass spec. So people, doing a check to see if it's just a sugar pill, say, oh no, there's artemisinin in there, but they don't quantify it, and selling that. So people are getting very low doses, and that actually breeds resistance. And so there's been a lot of effort to clamp down on that kind
What
of thing
about
as
the
well.
target?
What about the target?
What is the target? Does anyone know?
No, not as far as I know.
Is anyone working on it?
Not at Amyris.
Okay, well, thank you very much.